Philip, you're still muted.
So I hope it's now better. So it's my pleasure. Philip Hennecke is my name. I'm the director of the Institute for Infection Prevention and Control here in Freiburg, the host of this online course. Of course, this is a course on the epidemiology of infectious diseases, and infectious diseases are very well established as a leading cause of morbidity and mortality worldwide, and of course in Nigeria as well as in Germany. And depicted here, you just see emerging infections, specific emerging infectious diseases in the last 20 years. So this is a highly dynamic field with 60 to 80% of infections emerging from non-human sources. So obviously there's a lot to understand. A lot of tools are required to depict what's going on, to derive new diagnostic tools, and to really grasp the impact of these infections on your very community. And that's what this course is about. And I just want to highlight a little bit, this is data from the National Institute of Health in Nigeria, very recent data, where they analyzed the influenza epidemiology in Nigeria. And what you see here are the sentinel surveillance sites, 4 sites in Nigeria. You know the topography probably much better than I do. And this is the frequency of negative and positive results. And I think just looking at this interesting piece of data, it already shows you what you have to know and how meticulous you have to be to really understand what's going on. First of all, there are only 4 sentinel sites in your huge country, in the huge country of Nigeria. And how representative are these? How good are these? How much are the data that are generated reflecting what's going on in the states that are not having sentinel sites? And on the other hand, here you see, the bars together represent the total samples that have been derived, and you see huge differences over the years. So again, the data that you have for this year, how much are they really reflecting what's going on, and how much can they be compared to last year's if the sampling size is different so much between years? Another example is from South Africa. This is from Shabir Madi's lab in it's data on the Cape. And what they did, they were looking in incidences of group B streptococcal disease, so sepsis, in newborn infants, depicted per 1,000 live births. So it's relative numbers. And what you see, there are differences in incidence of at least 30 between these different regions. But that is very improbable to reflect what's really going on. And what you see here is that in Soweto, where there is a big hospital, they record many more cases than in an immediately adjacent region where there are few hospitals. So really showing again how important it is to analyze the data properly and to understand them and to calculate them. This is about numbers. This is about tools. This is about your knowledge that you are going to acquire during this course to really empower you to interpret this data and to communicate those, because they are, of course, extremely important for deriving consequences. Now, Nigeria and Germany, they are not so different in size. Nigeria is about twice as big in population, or more than twice as big in population and in surface area. But now when we say Germany, why Freiburg, or what's specific about Freiburg? And Freiburg, if you were lucky, you have already visited us, but Freiburg is really in the middle of Europe. The next big cities are across borders, Switzerland, France, in the southwest. And actually, this is an area where this is a knowledge area. This is a knowledge hub. The universities in Strasbourg, Basel, Strasbourg, France, Basel, Switzerland, and Freiburg, we are collaborating very closely, and they are all They are older than or about 500 years old. So this is really a knowledge hub here, the region that we are in. And Freiburg is an ancient city, as you can see here. It's a very beautiful city. It's an ancient city, but it has really cutting-edge technology, cutting-edge science. We are known especially for what we can deliver in knowledge with respect to infection and host responses. And what may surprise you is that Freiburg has the second largest hospital in Germany. So after the Charité in Berlin, we have the second largest hospital, which means, we have really the power to understand what's going on in populations. And we are, of course, as an institute, focusing on what's going on in a hospital. And we have a number of different research groups. We are an integrated center with covering all areas of infection prevention, infection pathogenesis, diagnosis, and even therapy. But today, we will really focus on two areas that we represent here, and this is infection epidemiology, quantitatively done by Chibbe, and with respect to disease done by Axel. And of course, there's this immediate overlap with global health. And with that, I will hand over to Chibbe again.
Thank you, Philip. And I will tell you something about the build-up of our course and what we will be teaching in more detail. So the goal of the course, as we said it, is to enable participants to use infectious disease epidemiology in their working environment and to assess the state of infectious diseases in such a way that it guides preventative and control actions and may inform public health policy in an impactful way. And basically what we're saying is we want to teach basic epidemiology, infectious disease epidemiology, that can be translated into action and therefore help you sort of design and inform, design strategies and inform policymakers. Now, to be precise, what the participants, what the students will know when they completed our course is they will have an understanding, they will be able to, sorry, there are people joining it. They will be able to understand current issues and priorities in the field of infectious disease epidemiology. They will be able to use research methods and understand how these methods can be applied to address particular research questions. And they'll be able to apply basic epidemiological, but also statistical methods, as well as social science methods to design epidemiological research questions for their hospital environment. And they will be able to use these skills related to prevention and control, management of infectious diseases, disease problems, and as well as health service problems related to challenges of infectious diseases. So that is basically how we set it in our course manual. Now, our course is a fully online course, and it will take 8 weeks. And what we'll employ is what we call a flipped classroom. There will be pre-recorded lectures, there will be study material made available, and there will be practical exercises made available for all the students to basically go through by themselves. And then at the end of each week, there will be a mentor session where the students can discuss the material with the mentor and try and sort of solve any problems that came up during the week. The total group will be split up into smaller groups of 5 students who during the week will sort of help each other in terms of any problems that arose. They will discuss what actually they would want to ask during the final mentor session at each week. One of each these groups will have to present on the material. So each week there is one group that will give a short presentation on what's been learned that particular week. Then by the end of the course, there will be that each student will individually give a presentation on which they will be assessed. This is on a topic of their choice, done individually, but multiple presentations, for instance, from a single group, can of course align. They can be joined in terms of one student giving one bit of the information and then next student adding to that. Sorry. So we have these first 6 weeks. First 6 weeks are the lectures, the practical exercises. These are things like Excel exercises, but also doing surveys and trying to analyze the data that you've collected yourself. And then there are these informal group meetings that I talked about, as well as the mentor sessions. Then the last 2 weeks, there are no further lectures added to the course, but these are basically meant to prepare for the final presentations in week 8. Students will be assessed on 2 main topics: student participation. So did you actually join in the mentor sessions? Did you go through the material? And the final presentations. They will then be awarded a pass or fail, no grading, no specific grading. In terms of these 6 weeks, the specific topics that we're going through is basically the understanding of the role of health staff in infectious disease epidemiology in the first week. Then the second week, there will be epidemiology and statistics, and the third week, infectious disease spread. And so these are the primary sort of topics that the students will need to understand to be able to formalize their epidemiological questions and do epidemiological research. Then we go into a number of examples. So we'll go through examples of how to set up surveillance systems, both normal surveillance, vector surveillance, and genomic surveillance. And we'll go through a number of example diseases in the 5th week, as well as outbreak response and prediction in the last week. So these are really the practical examples of what you can do with the of the epidemiological knowledge that you acquired in the first 3 weeks. And so why choose this Certificate of Advanced Study course? It's a unique course tailored to West and East African health professionals. We've got expert lecturers on each of the topics. So we have specific lecturers for each of the topics so that you learn as much as you can from them. There's a flexible learning condition. You can study the material whenever you want. Yeah, of course, you will need to be able to join the mentor sessions at the end of the week. But otherwise, there's quite a flexible working condition, a learning condition. And the University Medical Center Freiburg and the University of Freiburg are some of Germany's top medical and teaching institutions that have a lot of knowledge to offer. And with that, I want to hand over to Professor Axel Kröger.
Thank you very much.
Sorry, Axel, maybe to interrupt.
Yes.
Just for because many more people have joined. Maybe it's an occasion to display the QR code again briefly for 30 seconds so people can fill in the survey where they come from.
We have a link to us. So we have a survey for those that have just joined. We have a survey because we'd like to get to know you better. We want to sort of anxious to know who's actually joining this. And in the chat, there is a link to that. And I, at the moment, can't reach the chat. because I'm sharing.
I'm displaying the link every few minutes when I see
Yes.
that people join. And if there are technical difficulties, feel free to contact me. I'm here to help you with survey or with Zoom.
Good. Okay. Yes. Okay, Axel.
Okay, thanks very much, Tibbe and Philip. My name is Axel Kröger. I'm working at the Center for Medicine and Society at Freiburg University and at the same time for WHO in Geneva, taking advantage of the vicinity, which you have seen and been shown already. So Geneva is 3 hours away from us. And if you have meetings in Geneva, please come to visit us as well. My task today is to give you one example. Peter Sager will give the second one of the course contents. So I would like to introduce what happens in week 6 about epidemic preparedness or how to deal with outbreaks. Next. No, I'm still here. Sorry, the previous one. Because you see here a very colorful picture of very threatening and scary events. Outbreaks are happening all the time and are coming and going also to hospitals and other health institutions in Nigeria. And we think that hospital doctors and health staff in general should be aware of the threats and know how to deal with outbreaks. Outbreaks have a high cost in terms of money, but also in terms of human lives, and are therefore very important for the health services and the health management. Next one. In our lecture, this is the first box. We want to cover the following topics. We start with what is an outbreak and how does it differ from seasonal increase of cases. As you see in all climate-sensitive diseases, there is a season for diarrheal diseases, for malaria, for dengue, etc. But this is different from an outbreak, which is an unexpected sudden increase of cases. Why it is important to know about outbreaks, I mentioned it already because of the high costs. Then how do we know that an outbreak is coming when we want to prevent outbreaks? This means we have to predict outbreaks and use for this alarm signals. And we go into details when we talk in our course. But the most important part is to have a working and adequate surveillance system. We have touched the surveillance system already in previous weeks. Here we will strengthen the importance of surveillance. What is the purpose? What are the components? Active, passive surveillance, sentinel surveillance. What is the ideal and what is the reality? And then we come to outbreak management in hospitals, and finally to outbreak prevention and response. So these are the lectures which we presented. But then after this recorded PowerPoint presentation, which Tibbe mentioned already, we will give our students and participants exercises. Here are some examples, for instance, to develop a flow chart of the surveillance system in their own area. So they have to contact surveillance people and also look in the internet. Then, if possible, to do an interview with a focal point or points of surveillance in the hospital about limiting and favoring factors of surveillance, what recommendations they have, what are action points when you receive an outbreak warning, And what do you do when you have in your outpatient department a sudden increase of, say, diarrheal diseases? How can you study the source of infection? And then they learn how to do case control studies, simple case control studies, and the techniques have already been taught in the first week. And in the mentor session at the start, we will start always with a quiz, with an online quiz. And typical questions are, for instance, here on the right-hand side, how many outbreaks do you see in the diagram on the right? No, it's not only 3, but you see, in fact, 5. What is herd immunity? How do you define an outbreak? What represents the endemic channel? So all what is related to the topic which we will touch in this week. Next. Here you see a typical outbreak picture. In the colored part, you see the endemic channel, which represents the historical pattern of the disease. And outbreak would be if above the upper line of the endemic channel in this year or in this moment, cases are exceeding these. And this is shown in this blue line. This is a real outbreak happening, occurring, going very, very high. Now the question is, how can we predict outbreaks? And I mentioned already that we use alarm signals, and we are able to predict outbreaks about 5 to 12 weeks before they start, giving time to the health services to react. What usually happens when you talk about outbreak warning, this is meant early outbreak detection, which means when the outbreak has already started and the health services notice, oh, we have an increase of cases, unexpected, perhaps we have already an outbreak, but usually this is already very late for mitigating outbreaks. So in this we will go in more detail. Next. I mentioned already for outbreak prediction, outbreak detection, a functioning surveillance system is very important. Of course, everywhere the ideals are different from the realities, but it's useful to think about improvements. And a comprehensive system would be disease surveillance, which is perhaps the most common one, but also viral surveillance when you talk about arboviral detection. Then we have vector surveillance. We will have, with the help of a Nigerian entomologist, talk about this part. Syndromic surveillance, when you have little laboratory support. Climate surveillance, particularly for climate-sensitive diseases. Regional surveillance, if you have a change in serotype, for instance, for dengue, then you get an outbreak warning also for the whole region. And media surveillance, I think it is just starting in Nigeria, and we can think together what to do better. And then an important part is also the data collection. You have DHIS2 as a very common data collection tools for surveillance. You have the SORMA system in some states and others. So we will also think about how to smoothen and standardize data collection and data transmission, if it is paper-based, if it is digital, what are the delays to getting to the information to the state level and finally to the national level. Next. Hospital management of outbreaks. First of all, you have to be aware if there is a hospital contingencies plan. In some hospitals you will find it, but usually it is only in the desk of the director of the institution and not very well known by the staff. You have to learn different forms of triage, particularly in outpatient clinic, who goes to the intensive care unit, who goes to the general ward, and who can go home. And then dengue ward, malaria wards. This is a management when you have a real big outbreak, you have to make space. And there you need staff, you need supplies, you need beds or mattresses for protecting your patients against malaria vectors. You have insecticide-treated nets. And of course, everything is related to resources, particularly financial resources, and you need to plan early for an emergency fund. Next. Then risk mapping, also it's very important, particularly if you are at state level, you would like to know in which of my provinces or my districts or municipalities do I have high transmission areas and low transmission areas to know how to target with an outbreak alarm, but also usually interventions. And therefore, the risk mapping is very important. You can do it by putting the incidence rates of the disease on the map, or you can also compare the incidence rates with the average. So this we will discuss in more detail. Next. And finally, we will also talk about hospital-based control of infectious diseases. Here you have certain different options for dengue control. This is partially the same for malaria, partially very different. So for the different outbreak diseases, you have different interventions, and therefore we will probably split our group also in two parts. One will deal more with arboviral disease outbreaks, and the other more with like Ebola, Lassa fever, and these kind of outbreaks. This is a very short overview of what we intend to do. And now I hand over to Peter Asaga, who will give you the second example.
Okay. Good morning back in Nigeria and every other place in Africa. Basically, I'll just let you into a short overview of how the genomic surveillance, coordinates within the public health system. So next slide. So basically, genomic surveillance has emerged as a pivotal tool in the fight against infectious diseases and the management of disease outbreaks, especially in low and middle income countries, for instance, like West Africa, Nigeria, Kenya, Tanzania, where you have, these regions are often burdened by unique health challenges and high disease burden. So by leveraging on this advanced technology, public health officials can monitor the genetic makeup of pathogens, track their mutations, understand their transmission dynamics, and implement timely interventions, and it could equally help them, equipped with data for them to respond effectively in order to curb this emerging threat. For instance, a few weeks ago, we had an online meeting with the Federal Ministry of Health, and we realized that, the whole Nigeria, there was no arboviral data. So probably by enhancing this, we could have something that we can cherish on to begin to do good surveillance. Next slide, please. Yeah, so like I said, this next slide is basically almost same like the first one, just for early detection of potential threats. And when you have these potential threats, when you have a genomic surveillance or genomic activities in place, you can proactively monitor how these diseases, evolved into new variants and all of that. And this will give you an ample chance to, intervene swiftly and curb the threat. Next slide, please. So how do we, apply this in disease control? It provides health officials with real-time insight of the pathogen variants, identification, evolution, and mutation. By analyzing the genetic sequence of these variants, public health officials and researchers will be able to, control outbreaks and prevent wide spread of transmission, thereby mitigating strategies such as, targeted vaccinations, and putting in place travel restrictions and quarantine populations, and thereby mitigating the impact of, outbreaks like we could see during the COVID-19 era. So next slide, please. Yeah, next one. Yeah, so how do, genetic surveillance empower public health officials? Armed with genetic, data, public health officials can improve outbreak strategy. It can equally, give officials a broader understanding, of the epidemiology and patterns of disease outbreak. This will enable them, shape their corridors, for future resilience against disease threat. That's just the overview. Next slide. This is just the overview. We are going to go deep and open the capsid or the envelope of genomics, surveillance, and other diagnostics when we commence this program. But this is just an overview of what you should expect. Thank you. So,
okay, thank you very much, Peter and Axel. Before we go to the Q&A, I think it's time to look at the results of our Llama poll.
I would need to be unable to share my screen.
You are? Oh, wait. Now you are. Yes. Okay. So how many responses do we have? 5. Okay. So apparently we'll need to try jollof rice. I've never had it as a good dish. I see Peter already nodding. Okay, that's good. As well as moi moi. The others not so much. Okay, good. Our second question was which region of Nigeria are you from? Primarily the East and West. Okay. And in terms of languages, we see people speaking English, Yoruba and Igbo as primary languages. Okay. And then, so the primary area of expertise in healthcare that you're from, you are from medicine, public health, and community health work, all in equal amounts. That's good to know. And then, in terms of what you think as the most effective infection prevention practice, hand washing and vaccination, which is showing probably that you're also hospital based. And which type of health related events do you find most beneficial? Workshops, all of you. This is bad news for our webinar, but I can totally imagine that. Okay, thank you very much. And with that, I'd like to open the floor for any questions that you have. Or if any of the speakers want to add something to our to what we've already said, that's of course also possible.
Yeah, I would just like to add that this is, of course, a very much depending on your participation and your energy that will really bring this to life. Of course, I think we can assure you that the content will be cutting edge. This is really, you will get out of this course as a very different scientist and person and medical professional as to get in. So we can assure you that you will learn so much. But on the other hand, we really rely on you communicating with us to make this, bring this extra aspect of quality in. That always comes from us as colleagues working together, trying to solve problems.